Hello guys, welcome to Agriculture Tutorials. The current progress in the genes and networks regulating cotton plant architecture article. This research is taken place in China. Jiang Hong and Hu Li Bin Ma, authors of this article. Cotton is the most important source of natural fiber in the world as well as a key source of edible oil. The plant architecture and flowering time in cotton are crucial factors affecting cotton yield and the efficiency of mechanized harvest. In this model, plant Arabidopsis, the functions of gene related to plant height, inflorescence structure, and flowering time have been well studied in model crops such as tomato and rice. Similar genetic explorations have greatly strengthened the economic benefits of these crops. Plants of the Gossypium genus have the characteristics of perennials with indeterminate growth and the cultivated allotetraploid cottons. Gossypium hirsutum upland cotton, Gossypium barbadense C. Iceland cotton. Complex branching patterns. In this paper, we review the current progress, the identification of genes affecting their functional mechanisms associated with the branches pattern, branching angle, fruit branch length, and plant height. The focus mainly on plant hormonal signal transduction pathway, identification of plant architecture QTLs and pep gene family members, florigen anti-florigen systems. We highlight further research, modern mechanisms. The core aspects of the aerial plant architecture are determined by overall plant height, stem growth habits, and branching patterns, with peripheral structural reflects the size, shape, and quantity and relative arrangements of the leaf and flowers. The architecture of plant is the species specific, reflecting a strict genetic control. However, genes an important role in determining plant structure can be influenced by many exogenous factors such as light, temperature, humidity, and plant nutritional status. In the flowering plants, the shoot apical meristem and the root apical meristem contains large number of pluripotent stem cells that are maintained by continuous and periodic divisions from which different cell lineages are formed through which differentiation and continuously form new tissues and organs such as leaves, stems, branches, and flowers. In agronomically important crops, plant architecture has strong influence on methods of crop cultivation and management as well as design and efficiency mechanized plant harvest. Changing the flowering time enhance the ecological adaptability of plants. Changing the plant architecture can optimize the planting mode of crops. Therefore, flowering time and plant architecture traits are important goals of crop genetic improvement. According to Lang et al. 1977, first described the balance between positive flowering and negative flowering signals through grafting experiment in tomato and tobacco. The later they identified flowering gene like florigen and antiflorigen. Lang et al. 1977, McGree and Agree 2012, Richard and Lipman 2019. Florigen and gibberellin systems control flowering time and plant architecture. The occurrences of natural mutations in the system's exploitation or bioengineering efforts have led to crop improvement. For example, in the Green Revolution 1960, wheat varieties with short and steady stalks were selected in breeding programs so that crops could better resistant to lodging and weather hazards while maintaining a higher mutant. Scientists discovered the first anti-florigen molecule SP in 1998. Newly et al. In model plant species Arabidopsis thaliana, research into gene functions related to plant height, flowering time, and formation of branch and inflorescence structures have been studied extensively. Research on crops of agronomically importance such as tomato, rice, and the subsequent identification of gene targets for genetic breeding programs has greatly enhanced the economic benefits of the crops according to Wang et al. 2018. Now coming to cotton, cotton originated in tropical and subtropical regions where it showed indeterminate growth and perennial growth habit, and cultivated cotton gradually lose its sensitivity to photoperiod, mainly annual. Cotton plant architecture and flowering time are the key factors affecting cotton yield and the efficiency mechanization cultivation, because compared with the potential of higher yields per plant. Increasing the plant density has a great potential to increase the yield. In the regions of China, Xinjiang, Yuan, Autonomous region, the main cultivation feature of cotton is dwarf, dense and early, which has the made great achievements in cotton production in Xinjiang. The plant density in this area is generally 1,80,000 to 2,20,000 plants per hectare, making its largest and highest yield cotton planting area in China. According to C et al. 2018 and Zhan et al. 2021, cotton plant architecture is determined by many factors such as branching pattern, inflorescence structure, and plant height. This branching architecture is different from solanaceae plants such as tomato, Park et al. 2014 and Morris et al. 2019. The main stem of cotton displays monopodial branching, which maintains the characteristics of indeterminate growth and produces vegetative growth indefinitely. There are two kinds of branches in cotton. The basal vegetative branches are monopodial, shows indeterminate growth habit. The distal fruit branches show sympodial branches, and each sympodial unit is composed of internodes and a terminal bole with flowers, an opposite leaf and axillary bud that produce next sympodial unit, thus forming a pyramidal plant structure. Gore 1935, McCree et al. 2016, C et al. 2018. In this paper, we review recent progress in the identification and characterization of genes and quantitative trait loci, QTLs, regulating cotton branching pattern and branching angle, fruit length, plant height, and flowering time, as well as their application and potential in breeding programs to obtain beneficial agronomical trait in cotton. Greater understanding of genetic regulatory network of these progresses will lay the foundation for the future targeted bioengineering of improved cotton cultivars for higher yields and optimized cultivation and harvesting. The regulatory roles of plants, hormone signaling pathways in the formation of cotton plant architecture. Plant hormones play an important role in regulating meristem activity, which has profound influence of the plant architecture, according to Wang et al. 2018. The introduction of dwarfing traits into wheat and rice during the Green Revolution is now understood that we achieved the genetic alteration in gibberellin metabolism and action, according to Peng et al. 1999 and Sasaki et al. 2002. In Arabidopsis thaliana, the role of hormones and their signaling pathways in plant development has been studied intensively, according to Ongro and Lisa 2008. However, there has been comparatively little systematic research in the role of plant hormones and the underlying molecular mechanism in the regulation of cotton architecture and flowering. Nevertheless, some progress has been made through identification and functioning analysis of cotton orthologous genes important for hormonal signaling and their targets in mode plant systems. Signals derived from several factors, including hormones, nutrients, and environmental factors, cooperatively regulate the growth of buds, and their regulator signal pathways conserve into branched one BRC1, means capital BRC1, which ultimately determines if the auxiliary buds are dormant or growing. BRC1 gene encodes TCP transcription factor, which is specifically expressed in axillary buds and participates in inhibition of bud growth and branching. Bhad et al. 2002. Mage TB1, capital TB1 in is ortholog hormone of BRC1 in Arabidopsis thaliana, which has been studied intensively for regulating role in branching patterns. These regulatory roles are in branching patterns studied by Hubbard et al. 2002 and Wang et al. 2018. GHBRC1 is highly expressed in axillary bud tissues of cotton with nulliplex branch, means no fruiting branch phenotypes, and is induced by various hormones. Sun et al. 2021. The overexpression of GHBRC1 in Arabidopsis resulted in a significant reduction of rosette leaf branch number. The video is going to higher duration, so this up to this is enough for the knowledge of cotton cultivation to plant architecture of architectural changes and the plant network gene regulating models in current progresses of recent years in China and various countries globally. Cotton is an important source of natural fiber in the world as well as source of edible oil. Thanking you friends. Have a nice day. Please like and subscribe to my YouTube channel. This is also my PhD research work. So I am studied this article, cotton plant architecture. This some articles are introduced in my PhD thesis also. The Wang et al. And all other members 2018 he supported very well in my thesis article. This article is used in my thesis sources. All the important concepts are taken by various articles. It is used for cotton plant architectural studies. Thanking you friends. Have a nice day. This source is taken by Frontiers in Plant Science. This is just for giving a awareness to my subscribers because I am explaining my cotton in cotton plant architectural models and thesis in various events. So I am explained this video. Thanking you friends for attentive listening and patience. Jai Hind.